So Carl Dieffenbach has had a very good year. He joined National Institute of Allergies and Infectious Diseases in 1992 and really had been an architect for the basic science division, which he headed for a period of time. He worked first on the vaccine trial, helped establish the Vaccine Trials Network, was in charge of the basic science division, and took over in 2002 the Division of AIDS Research at NIAID, National Institute of Allergies and Infectious Diseases. And since that time, realized that every result you heard from the Capri study in 2008 to now was initiated, the leadership and architecture for it came on his watch. And I have to say, when I first read his article in JAMA, was it 2009 on test and treat? And the specific strategy I had a lot of problems with. But the idea, how the implementation was proposed for this specific strategy was debatable and still is. And we've heard a number of different perspectives on that today. But the idea that we need to test and the idea that we need to treat was the core belief, the core strategy that the data over the last 2.5 years have consistently confirmed. And that then was followed with treatment as prevention with Tony Fauci and Carl Dieffenbach a year later, has really changed the discussion, the dialogue, the possibilities in a fundamental way. And we're lucky enough to get to hear Dr. Dieffenbach this afternoon on his next round of architecture and strategies, next steps on treatment as prevention. Carl?
Thank you, Mary Jane, and thank you, Tom. It's a pleasure to be here and participate in this meeting. What I'm going to try to do is give you a sense of where we have been and where I think we can really utilize the tools we have and develop some new tools that will really have a profound influence in not just the domestic epidemic, but the global AIDS pandemic. Mary Jane wanted me also to stress this from this presentation from the perspective of dealing with our more targeted, our more general, our more challenged populations in terms of substance-using populations and the mentally affected. So I will try to weave that in, and if I forget, I can count on Mary Jane to ask me lots of questions about that during the question and answer session. So if we think about the spectrum of opportunities to impact on HIV, we have now filled in specific activities in every single one of these cells. We know we have behavioral interventions, structural interventions, and adult male circumcision that has been shown to be quite effective in unexposed individuals. We have PrEP and microbicides. We have PEP. We're working on an HIV vaccine. And then we have, within this past year, absolutely proven, beyond a shadow of a doubt, that treatment that reduces HIV infectivity has a profound influence on HIV transmission. So moving forward, what do we do with all this information? Well, in order to really control an epidemic or to help prevent new infections, we are still in a situation where no single strategy is sufficient to really tame the HIV AIDS epidemic. Therefore, we need to develop a toolkit, keep this toolkit refreshed and up to date, and at the same time, link the activities within the toolkit to the specific epidemic and population we're trying to tackle and target. And clearly, until we get to a situation where we have an HIV vaccine that is 100% effective, like a smallpox vaccine or a polio vaccine, or some other strategy, we will absolutely have to maintain the toolkit. Even at that point, we will still need the toolkit because there will always be a behavioral component in everything that we tackle, not just in this disease, but in medicine as well. It is not sufficient just to have the tool. You have to figure out how to get people to take the tool, come back for appointments as needed. and basically engage in the discussion with their healthcare provider or their prevention team about how they're going to take advantage of the activities that are available to them. So just to spend a moment on that point. So I think that if we really stop and think about where we are in the AIDS pandemic 30 years into this infection, we are in a process of transition. And it has sort of come upon us like a small but steady high tide. And where we are today is we are in the throes of a transition. And the transition we're in the throes of is from managing an infectious disease to managing a chronic disease. And I think that is one of the things that we're struggling with as we treat HIV in many ways as an infectious disease, appropriately so. It is an infectious disease. But once HIV infected and once identified as HIV positive, it's a new set of tools that we have not fully embraced. There are some places that have been better about this than others. But I think that is our long-term challenge, is to figure out how to make this transition in an effective way so that we can maximize the tools that we have available. And I'll come back and talk a little bit more about that at the end of my presentation. So it has been a really good run of 2.5 years or so. And I think that the challenge will be next year. It'll be, what have you done for me lately? So we'll see what kind of press the division and the Institute are getting in a year. But it really began with the Capri, with the RV144 trial that showed a very modest benefit of an HIV vaccine. And we're in the process now of analyzing that data set to see if, in fact, we can get a glimmer of a correlate of immune protection. Moving on to September of 2010, we had the results of the Caprisa trial. And again, just to point out the challenges that this gel has. It had 39% of effectiveness overall. In high gel adherers, it was 80%. But the lower bound on the overall trial was 6%. So this is an example of a product that, if used properly, could have very high efficacy. Used improperly, could potentially be extremely problematic. Moving on to November, we had the PrEP trial that was run out of UCSF, Bob Grant's IPREX trial, enrolling almost 2,500 seronegative men and transgender women around on 3 continents. So this showed a 44% reduction in HIV incidence, again, with a profound reduction in people with high adherence and nearly flat in people that had very low adherence. So again, tremendous influence of behavior and uptake with this trial. So the next 2 trials I'm going to talk about are in many ways very contrasting, because what the Gates Foundation and USAID had tried to do was in many ways start a trial that replicated Bob's IPREX trial in women, and they called it FemPrep. This trial was stopped for futility. There were 28 infections in each arm, the treatment arm and the control arm. Why? Is it all adherence? Is there biological component to this? This is under active study. And this actually, again, reinforced the need to make sure that where possible, we link behavioral and biomedical. At the same time, this population was extremely challenged. This was a very difficult, relatively poor population in southern Africa, Kenya, and Tanzania. Many of the women did not even have enough money or enough, they didn't even carry a purse. Many of them were not commercial sex workers, but traded sex for food. So how can you expect somebody to take a pill a day if they can't even eat? Or they had other problems in their lives. So this is really getting at the issue of managing the patient as a whole and not just dealing with the disease by itself. We had the result coming out at the International AIDS meeting in Rome of the CDC Partners as Prevention trial, where in many ways this was the best possible outcome, because this was a couples study where essentially the team enrolled couples and they followed couples, and couples came in for counseling, motivational counseling. In studies done by the Harvard group, looking at pill use 3 ways, the level of adherence was over 95%, and the level of activity in that trial was also significantly showed significant efficacy. The CDC trial also presented their results of their TDF2 trial in Botswana, which also showed a positive signal. Now on to Mary Jane's test and treat. I mean, I think that when we wrote the commentary for JAMA, we wrote it from the perspective of, while this is a good idea, it's in many ways a highly utopian approach. But there are some significant research challenges that need to be addressed in order for us to truly understand what it will take to have test and treat or treatment as prevention really work in a real-world setting. And there were a couple of absolutely key questions that we felt needed to be answered. One of which was, does in fact treatment of an HIV-positive person reduce transmission to a seronegative partner? And the way that we had decided to pursue that question was using a couples design, and the trial was called HPTN-052. We enrolled almost 1,800 couples, and we were able to show that, in fact, in this design, that between 96 and 98% reduction in HIV transmission, when you could track the transmission between the infected partner and then the individual who became infected. There were a number of infections that occurred outside of the serodiscordant couple. And those were, that just proved once and for all that, in fact, this reinforced the message that this actually works, because if you're going to have sex with somebody, actually you're at risk. And it doesn't matter whether you're in the discordant couple or not. But the point was here, the drugs, the antivirals did have a profound influence on linked transmissions. As we look at the future for this, well, we have this kind of a cascade. And this is actually an impressive set of studies that have been done, from FemPrep, where we had 0 efficacy, all the way up to ARV as prevention, in terms of the step-ladder effect, in terms of what we're seeing for effect size. So what we have in this situation is a large number of trials with relatively large confidence limits that really are affected by social, behavioral, and other consequences. So this is in many ways the challenge of the next range of trials. How do we make this a little bit more generalizable? How do we take not just the biomedical side, but the behavioral side, and improve the efficacy and narrow these confidence limits? So a trial that we have ongoing will absolutely finally answer the question about PrEP and vaginal microbicides in women. This is a trial that is now fully enrolled in South Africa. It's called the VOICE trial. It has the following relatively interesting design, where there's essentially 2 arms, an oral arm and a topical arm. And the topical arm will support or refute what was seen in Capri and provide the safety data needed for the licensure of tenofovir gel. And then the oral arms will really help to nail down the question about whether Truvada or tenofovir is sufficient, or whether tenofovir is sufficient, or whether you need Truvada to really see a PrEP effect in women. So in terms of the future of what we're doing in pre-exposure prophylaxis, there are a number of demonstration projects that are in the works or being planned. The IPREX open label extension study, which also goes by the clever moniker of IPREX-Olay. And keeping in mind that this trial was done heavily in South America, it's actually very clever marketing on the part of Bob Grant and the colleagues at UCSF. And this really is to assess, now that we have a sense of the benefit of PrEP, will adherence improve in these target populations? In a sense that this is a well-characterized cohort, and in many ways, many people may say that, well, it's a contaminated cohort because it's already been enrolled in a clinical trial. But it's a first step toward understanding how to roll PrEP out. Working with the San Francisco Department of Health, we're in the process of putting in place a PrEP demonstration project. And then we have plans for additional PrEP demonstration projects in many of the 12 cities where we are working on the President's National HIV/AIDS strategy that will come into play over the next 5 to 6 months. In terms of next steps on PrEP, there's a number of studies that are in the pipeline or have just initiated, several of which are looking at just answering some of the details and questions around the use of tenofovir and Truvada in terms of the pharmacology and whether or not we can develop an alternative dosing scheme other than daily. And that's what the goal of HPTN 066 is, to really get us good pharmacology and pharmacodynamics data on these two drugs in a number of men who have sex with men in 2 sites, one in Bangkok and one in the United States. 067 is a partner study, a parallel study that's linked to 066 that really looks at the behavior of pill taking and really looks at the question around intermittent PrEP. So we feel these are important trials that will really add to our understanding of what PrEP entails. And then we need to start thinking about what are the next generation of studies that need to be done. What are the next products that need to come into play that will be better than tenofovir, safer than tenofovir, and move us closer to agents that really could be utilized by larger numbers of people. For those of you who truly understand this, I mean, tenofovir and Truvada have a challenge in that people with kidney disease can't really utilize PrEP. And so some of these newer agents, such as Maraviroc, will make a difference. So we have a study called 069, which is co-sponsored by the AIDS Clinical Trials Group and the PTN to look at the safety and tolerability in a phase 2 randomized double-blinded controlled 4-arm study of 4 different PrEP regimens. Again, no placebo involved here. So in terms of other studies we've got on microbicides, we're considering a trial very similar in structure to the Iprex-Olay trial, the follow-up from VOICE, and that will be called the CHOICE study. And that will, if both PrEP and a microbicide are shown to be effective in women, then women will literally be given a choice, and they will actually be able to switch back and forth for a 3-month window to see which they like better, which fits better their lifestyle, a ring, a gel, or a pill. And then ultimately they'll need to lock down on one of the two, and then we'll follow them for a period of time. Additionally, if you think about the challenges with a gel, particularly one that is coitally dependent, are there ways to help affect the behavior by essentially creating a device that will deliver the antiviral to the site of action in a way that will put it in, and so behavior is minimized. So basically, you insert the ring once a month and then change it, and therefore you're not having to remember to use the product in a coitally dependent way. Additionally, we're continuing safety studies on tenofovir in order to complete the licensure package for tenofovir gel. So to look ahead, and this is sort of the blue sky portion of my talk, where we're going to talk first about the next steps of what we need to do in the relatively short term. We need to complete the VOICE trial to get tenofovir gel licensed. Now, the interesting part of the tenofovir gel story that is also moving forward with not just its HIV prevention component, but it also, in the Capri trial, had a profound impact on herpes. So we may end up with a product that gets licensed here that not only can prevent HIV transmission, but also herpes transmission. And that would be a really good double benefit from a product like a gel. Additionally, we're moving forward in additional adherence studies and trying to figure out how to improve the uptake of not just the gel, but also of PrEP. We're continuing discovery and development of next generation for both these areas, and we're particularly focusing on the development of rectal microbicides. This is an open area of research. Don't know if, we know PrEP works quite well in men who have sex with men. Will a rectal microbicide be as good or better, or at least adjunctive? Time will tell on that. Also, as all these products come forward, we need to think about how they can be utilized within a comprehensive prevention package and define the adherence strategies, delivery systems, and social marketing approaches to make them more universally available and get us to a point where they are evaluated in combination prevention approaches. And ultimately, the area where I think we're going to be struggling with at the NIH for a while is how do you use placebos in the future in prevention studies now that you have all of the success? So I think that that's an ongoing debate, and there's a number of consultations planned with in-country as well as with ethicists in the developed and developing world. Now, to switch gears a moment and talk about this research agenda that needs to go forward with treatment as prevention or test and treat. There are some significant challenges we face in implementing this strategy. And how do we go about testing? What are the behavioral barriers to testing? What is the relationship between stage of HIV infection and efficacy of transmission? We've proven efficacy of ART in preventing transmission, but at the same time, there's a major issue here in that starting therapy early, is it truly of benefit to the individual who's starting therapy, or is it solely a public health benefit in terms of impact on transmission? And ultimately, all of this comes down to cost effectiveness for society. So if you think about this cascade, and this is, I think, the thing that troubles most of us in this room, this is actually, if this were a report card, it's actually a terrible report card. So in a given population of 100 HIV-positive people, at the end of the day, we as a society in the United States are capable of identifying approximately 79 of those 100 people who are HIV infected, link then 47 of the 79 to care, and get about half of them, of those individuals who we link into care, sustained in care, virally suppressed, and controlling their HIV infection. So this is, I think, the challenge that we all face. And this is where those populations of the substance-using populations, the people with mental health challenges really comes in. Because we need to tackle this in a different way. Right now, when people test, there's a whole range of ways people link to care. Sometimes they're given a phone number. Sometimes they're actually tested in a facility where the linkage to care occurs. At the same time, you can't just manage people's HIV disease. You need to manage the whole patient. And somehow, we as people responsible for implementing the President's National HIV/AIDS strategy need to acknowledge that, and need to acknowledge that we need to change many of the things we're doing, particularly in the area of linkage to care. So if test and treat or treatment of prevention is going to have that kind of an eye-popping effect that Mike Cohen showed in HPTN 052 of 96 to 98% inhibition of transmission, we're going to have to change this number so that when we're talking about 100 people who are HIV infected, that the number of people who are virally suppressed and in care and in sustained care is significantly more than 26. We're going to have to bump that number up to 80, 85. And that is really the challenge we face. Now, there remains a dynamic tension in this field. And the tension I feel, particularly in the areas of PrEP and microbicides, is now that we have some success, what do we do with this? Do we optimize what we have in terms of optimizing Truvada or tenofovir, or do we move on to the next generation? And to my mind, I think that this tension is healthy, and I think it requires a level of discussion. If you take time to take tenofovir and move it on from a gel and make a tenofovir ring, make a ring of tenofovir that then also has a contraceptive form, it will be as good as the level of tenofovir that you can deliver. At the same time, can we move on in other ways and develop other long-acting formulations that improve treatment outcomes as well as prevention? I think that this is one of the areas that excites me most. That is, not only can we make rings and implants and injectables, but we're also working on long-acting oral formulations. So in terms of a way to deal with this is, can we develop a treatment armamentarium where essentially HIV management is simplified? And right now we're pretty simple. We're down to one pill once a day. Can we take a leap and move to a situation where we're dealing with not ART on a daily basis, but ART that is delivered in 4 to 12 doses a year? In that situation, you could then have the linkage appropriately where the least of the individual's challenge was their HIV infection. You could then manage their substance abuse. You could manage their mental health needs in a way that wasn't complicated by HIV treatment. At the same time, if we move forward in this area for treatment, we can also get to a point where we have PrEP, or a microbicide, ideally PrEP, that was also deliverable in terms of 4 to 12 doses a year. Now, if you think about the challenges that somebody like Ambassador Eric Gooseby faces in terms of trying to deliver one pill once a day to 6 million people around the world, if he could simplify that and deliver 4 doses a year to 6 million people around the world, his life would be simpler. Over time, I think cost would go down, adherence would go up, clearly. At the same time, we could really start to then have a conversation about how to combine this kind of an approach in combination prevention and combination treatment, where we could manage each patient for their own unique characteristics. So at the end of the day, we have a set of challenges that we face. And if you look around this ring of effective and both proven and researched prevention strategies, many of them these are so behaviorally dependent and require this type of focused study. But at the end of the day, if we can come up with some very exciting new tools that will help us manage that component of the HIV disease, where we can then focus on education and on handling the individual nature of the individual patient, so we're managing this as a chronic disease, I think we can get to a point where these tools will really begin to control the AIDS epidemic, and we can then move toward essentially thinking about controlling and hopefully eliminating the AIDS epidemic in our lifetime. So that remains our goal. It's not easy. The conversations that we were just listening to around ADAP and the realities of the strength of the United States is we're a group of 50 states. I suppose the weakness of the United States is also when the state law comes into being, we're stuck with 50 states. But ultimately, we all need to work together and tackle this problem and get to a point where we can control and ultimately end the AIDS pandemic. Thank you for your attention. Okay,